All right, so your next class with me is your unit test on ATP and respiration. And the only thing we haven't done is talk about the aerobic steps of respiration. So that's what we're going to do today. So I don't know Pear Deck, nothing like that, but I'm going to give you some drawings and some notes and talk you through some of the tougher parts of this, of these complicated reactions, and try to make it so they don't seem so huge, okay, and overwhelming. So the first thing that I'm going to do is I'm just going to take you back through what we already did, right? So remember that cellular respiration, cellular respiration can be aerobic or anaerobic, and we did those anaerobic steps, right, with no oxygen. We already did that, and let's just quick review anaerobic respiration, right? Okay, so what's the first step of anaerobic respiration, and actually the first step of any respiration? Tell me what it is. Come on. Glycolysis, excellent. Glycolysis is the first step, and where does glycolysis always happen, no matter what type of cell it is? Where in the cell? Yes, in the cytoplasm. Beautiful. All right, in the cytoplasm, and that is true whether you are doing anaerobic or aerobic. All right, the second step of anaerobic respiration, once you have done glycolysis and you have your pyruvates, what happens to them? What's the next process if you don't have oxygen? You start with glycolysis, you don't have any oxygen, so the next step, whether you're in a bacteria or yeast, or whether you're in one of your skeletal muscle cells, the next thing that happen is fermentation. Yes, very good. Okay, and fermentation also happens in the cytoplasm, by the way. Remember, fermentation is something that that you're only doing to replenish the NADHs. Remember that? So I mean, there's two types of fermentation. There was the alcoholic type that happens in the yeast cells, which is why we use yeast to make our wine and beer. And then there's lactic acid fermentation that happens in your cells, your skeletal muscle cells, when you're exercising a lot and your cells have to run out of oxygen and they have to turn to a form of fermentation. So there's anaerobic respiration. That's that part. And we have done that. We did that already. So now let's go over here and see what you know about aerobic respiration, okay? So now with aerobic respiration, again, what's our first step? No matter what kind of respiration we do, the first step is always glycolysis. Yes, very good. All right, so glycolysis is still going to happen with all the very same enzymes as first evolved in the very first little tiny bacteria cell, right? Glycolysis in the cytoplasm is still happening, even in a complicated cell like one of your muscle cells or one of your brain cells or in a plant cell. Eukaryotic cells do this too. Okay. Now the question is, once you've done glycolysis and now you're in a eukaryotic cell that has a mitochondria in it, now what happens instead? If you have oxygen and you have a mitochondria, what's the next step? It's a step that was named after a German gentleman, Hans Krebs. Yes, okay, so we have the Krebs cycle. It's also called the citric acid cycle. And that happens in what cell organelle? You guys are really sleepy today, aren't you? I know you know it. Mitochondria, okay? Happens in the mitochondria. Yes, Hannah and Reese, beautiful job. All right, Krebs cycle in the mitochondria. And then the last step that really gives you the biggest payoff of ATP, when you have oxygen, is the electron transport chain. Yes, electron transport chain, which also happens in the mitochondria. There, all right. So this we have not talked about the Krebs cycle and the electron transport chain, which is going to go into more detail today. Before we do that, what is the biggest, most important payoff of aerobic respiration, and how many of them do we get? Yes, 6 ATP is a very big payoff per glucose molecule of ATP, and really can't happen without all the specialized membranes and proteins that are present in a mitochondria and not present in the cytoplasm. What about over here? What payoff do we get from fermentation and glycolysis alone? Remember the net payoff of ATP? 2. Beautiful job. So much less, but if you're a tiny little bacteria cell or yeast cell even, you can manage with that much. But us, with our expensive organs and our brains and everything, we need a lot more. Okay, so there's the bird's eye view right now. This is what we've done. We did all of this already. We talked all about this, and we've talked about glycolysis pretty thoroughly. So this is where we are today. We're going into the Krebs cycle and the electron transport chain, and we're going to find out why there's such a big payoff with that. Okay, let me erase this. The next thing I want to do is I want to give you a couple more, a couple things before I go charging into a big drawing of the mitochondria that I'm about to do. I want to give you a heads up on some redox reactions that are really important and that are not necessarily easy to understand if I lay them on you when I'm talking about a bigger process. I'm going to look at them separately just for a minute. So if you remember your chemistry, redox reactions, right? You might remember something called Leogur, or you might remember something called oil rig. I don't know if I'm spelling that right. Oil rig. The one I learned was Leogur, which means that, just very simply, when something is losing electrons, that is oxidation, being oxidized, right? So if something loses electrons, it's being oxidized. And if something is gaining electrons, it's being reduced. Okay, so that is kind of the translation. That's what Leogur means. I'm sorry, that's the one I learned. If you learned the other one, that's fine, as long as you can keep track of it. Okay, so really, what's really going on with all of these respiration reactions, this huge metabolic processes with all these arrows and things that you're going to see, it's as simple as this. You're starting with glucose, right? You're starting with glucose molecules, and little by little by little, right, step by step by step, regardless of whether you're in glycolysis or whether you're in the Krebs cycle or whatever you're doing, you are breaking that 6-carbon molecule into smaller and smaller and smaller bits and releasing the energy from this glucose molecule little by little by little. So what you have to think of is this. There are a bunch of high energy electrons sitting in all the covalent bonds inside that glucose, right? All the covalent bonds holding that glucose molecule together are made of electrons. And what you're really doing here is you are taking, every time you break glucose from if we break glucose from its original 6-carbon, and then here, somewhere down here, you've got, at the end of glycolysis, it turns into 3 carbons, and then it goes to 2 carbons, and then it's down to single carbons because carbon dioxides are coming off. Every time you do that, every time you break the glucose down, you're releasing electrons. You're releasing electrons every time, right? When you are breaking those bonds, right? So the flow of electrons is coming, and really energy, really, right? The source of energy for really making all those ATP is coming from an electron flow. And we haven't really talked about that yet, but that's really what the truth is. So you're getting electrons that keep coming off and coming off and coming off, right? And those electrons are being picked up by 2 electron carriers. What are the names of those electron carriers? There's 2 of them. There, you do the acronyms. Just do the acronym. NAD and FAD. Beautiful, Kevin. Yes, that's it. Okay. NAD and FAD are both electron carriers that they're the same sort of rechargeable batteries as ATP, except that instead of taking phosphates on and off, they're taking electrons on and off. So let me type this over here. Every time an NAD plus gains electrons and is reduced, it becomes an NADH, right? Oh, what just happened? Right, there we go. So those electrons coming off here are picked up by NAD pluses. They can also be picked up by FAD. I'll just make it simple. FAD become FADH. Oops. 2. It's been reduced, okay? So both of the reactions down here are reduction reactions where those electron carriers are picking up electrons that are coming out of these bonds that are broken in the glucose until you get smaller and smaller and smaller and you don't have anything left to break down, right? And down at the bottom, you have the ultimate products down here, which are carbon dioxide and water, right? Oops, water. Okay. Carbon dioxide and water are worthless. They're waste products. They're the products of this reaction, but they're not doing anything energy-wise for you, right? They are actually waste. Carbon dioxide is a waste you have to get rid of. So remember that the point of respiration is not to make these guys. Yes, they're your products. Yes, they're the end product of breaking the glucose down into small bits. But the real prize are these electrons coming off and being picked up by NAD plus is picking up this electron here to become an NADH. Right? So the flow of electrons is going from the glucose to all these NADHs, and NADHs are the main thing that are picking these things up, right? And then way down at the bottom, when you're in the Krebs cycle, you're also picking up. You're also taking these guys and putting them into FADHs also, which are the other type of electron carrier. FADH, right? There. Make it complete. Sorry, I'm just going to make it neater. There, right? Okay. Okay. So, I mean, this is just a bird's eye view. We're going to go into more detail in just a minute. But basically, the aerobic respiration reactions are harvesting electrons all along the way. The anaerobic respiration, don't bother with that. The anaerobic respirations are not collecting high energy electrons. They are making ATP directly using enzymes and substrates. But the ATP making processes in the aerobic reactions are very different. They're using these high energy electrons. Oops, where's my drawing? This guy, and this guy, and this guy. And there are a lot of them. A lot of these guys carrying these high energy electrons are going to go to the place, which sounds logical for them to go, towards the electron transport chain, which is in the mitochondria. And that electron transport chain makes use of those high energy electrons to take ADP molecules that are already floating around inside the mitochondria and phosphorylating them into a whole bunch of ATP molecules, which are the most important product of these reactions, right? That's where the money is. And the flow of those electrons through that chain, I'm going to show you where that is in a minute, is harnessed. That energy flow is harnessed to create a force, a diffusion force, that influences the phosphorylating of ATP. That's not going to make full sense until I show you the bigger picture here. But that's basically what's happening. You are oxidizing glucose little by little by little and making all this carbon dioxide and water at the end, which is worthless. But every time you oxidize glucose or intermediate sugars, you're taking those electrons and reducing electron carriers. You're putting them in a molecule that's carrying them. Can't see my hand. You're taking these electrons and reducing them. So now they're carrying these high energy electrons over here to a special place in the mitochondria, which uses those high energy electrons to phosphorylate ATP. And the final home of those electrons, and the whole reason for oxygen in the first place, is once their energy is used up, those electrons, all those electrons get picked up by the final electron acceptor, which is oxygen gas. This is why oxygen winds up being so important, why we call this aerobic respiration in the first place. Oxygen's ability to pick up those electrons at the end of their job, when their job is over and they have helped make all that ATP, and eventually that's what makes the water. Is the final product up there once the oxygens have caught those electrons, is they eventually make water. That's what makes all that ATP making possible. Okay, so hopefully that kind of gives you a better idea of what I'm about to explain. All right, any questions yet? Shall I keep going? I'm going to assume that's a yes. Okay, I'm going to erase this. It's all recorded. It's all recorded. All right, don't be falling asleep. Keep paying attention. Okay, test is next class. You need to know this. All right, so here's the choice. I have a worksheet open that I could draw on, but it was glitching last time a lot, and it's been glitching all morning. So I think I'm just going to draw it here. Is that okay with everybody if I just draw it right here? Is it big enough? Yes? Okay. All right. Just keep it right here. That way you won't have it go green and rainbowy. It's been doing that. So we'll just keep it right here. All right. So what I'm going to draw here is a giant mitochondria. I'm going to draw a giant mitochondria, which really, if you think about it, a mitochondria is just an ancient aerobic bacterium that went endosymbiotic and got inside our eukaryotic cells and started making mucho ATP for us. It's kind of a cool thing to think of a mitochondria as a living organism within our cells. Kind of creepy, but kind of cool at the same time. Okay. The mitochondria, and mitochondria have two layers. They have two compartments, actually. So the outer layer is predictably called the outer membrane, and then there is an inner membrane, and then here there is a, what do they call it, and intermembrane space, which turns out to be really, really important. And then deep inside here, this region is actually called the matrix. Inner compartment of the mitochondria is called the matrix. Okay. And out here, remember, out here is the cytoplasm of the cell, right? Out here. That's what that is. All right, so let's talk about what we already know. We know that out here in the cytoplasm, glycolysis has already happened, right? Glycolysis happens here. And with glycolysis, we are taking glucose and we are breaking it down until we get pyruvate, right? So 2 pyruvates are the product of that, right? And from that, we get a couple of things. We get a little bit of ATP. So you get 2 ATP from that. And you also get, don't forget this is important for the accounting, you also get 2 NADHs that are reduced from this, which can be used by the electron transport chain and can add to our ATP total in the end. All right, so we know that. We already did that glycolysis thing. Now those 2 pyruvates are going to come all the way into the mitochondria, right there. And they're going to go through something that if you looked at my video already, you know is called the link reaction. It's kind of a reaction between glycolysis and the Krebs cycle, which the book will tell you about and I will tell you about. And it actually is important because there are some things happening with it that add to the ATP as well. So this is called the link reaction. And in the link reaction, you're going to take off another carbon. So remember, pyruvates have 3 carbons, right? 3 carbons each. And what happens here is one of them is lost and becomes a first carbon dioxide. So your first carbon dioxide gets made and you lose a carbon. Right. So now you have a 2 carbon molecule left, which is an acetyl group, an acetyl group, which is a 2 carbon molecule that hitches itself to a very, very special enzyme called coenzyme A. Okay. And that is a 2 carbon, a little 2 carbon group is all that's left of the pyruvate. But that's okay, there's still energy to be invested there. Okay. The other thing is when we broke that carbon dioxide off, something else came off. You also reduced another NADH here. So you made another NADH here. Actually, you made 2 of them, because there's 2 pyruvates, and in fact, we made 2 carbon dioxides too. And that's the link reaction. The pyruvate, those 3 carbon pyruvates come in, and they get a carbon broken off of them to become a 2 carbon acetyl group, right? And when that carbon dioxide is broken off, remember those electrons got released because you broke them, and those electrons get picked up by NADH. So you have some reduced NADHs there. Okay. And now all that's left of your pyruvate molecules are these 2 carbon acetyl groups. But that's okay because we're going to put them into something called the Krebs cycle. The Krebs cycle is very often represented by a circle like that. It goes round and round. So Krebs cycle. There. Okay. So here's what happens. This little 2 carbon group has kind of this coenzyme that's attached to it. This coenzyme A is kind of like a big brother that grabs you by the hand and pulls you into the party with the big kids. And that's what the Krebs cycle is. It's like the big party with the big kids. And in order for that tiny little 2 carbon group to find its way into the right place, that coenzyme A has the ability to escort it in to meet the molecule it needs to meet up with to join the party. And the molecule it's meeting up with is a 4 carbon molecule that's right here. 4 carbon. You don't need to really know the name of it. 4 carbon molecule meets up with the 2 carbon molecule and becomes 4 plus 2 is 6. A 6 carbon molecule. It's actually called citric acid, hence the citric acid cycle name. Sometimes they call it that. Okay. So then what happens is that 6 carbon, if you just trace the carbons, it's easier. Certain things happen. You get a carbon dioxide that comes off, another one, right? Another carbon dioxide comes off, and then that becomes a 5 carbon because it lost a carbon. And then another carbon dioxide comes off at some point. And now you're down to 4 carbons again, and then it makes, and then it comes around and meets up with the next acetyl-CoA, right? That's why it's cyclical. Remember, it's cyclical. So that 2 carbon thing comes in, meets up with the 4 carbon, becomes a 6 carbon intermediate. The next enzyme takes it, breaks the carbon dioxide off, makes an NADH. So at the end of all of this, every time carbon dioxides get broken off, every time bonds get broken, you are reducing those 2 electron carriers we talked about, right? You already understand that there are NAD plus and FAD pluses floating around in here, waiting for those carbons to be broken off so they can catch those electrons and be reduced into their useful states, NADH and FADH2. Oops. But you have two electron carriers that are carrying high energy electrons, and that was the whole point. Now, somewhere over here, there are also one particular special enzyme that helps to phosphorylate a single ATP. So you get one little ATP out of this, only one. Every time it goes around, the Krebs cycle phosphorylates one ADP into an ATP. One, that's it. But you get a whole bunch of NADs and FADs that are reduced and carrying those electrons. So let me put a little bit of color in here. Okay. So the important guys, the NADHs that have been reduced and are carrying those high energy electrons that they got every time carbon dioxide or bond was broken and electrons were released into them, right? Look at all the electron carriers we get. All of these guys that are carrying high energy electrons that are now predictably going to go over to something called the electron transport chain. Okay, so what the heck is an electron transport chain anyway? What in the world is that? That's what I used to wonder when I was sitting in your chair and I was trying to read the book and understand from the pages of the book and from what my teacher was explaining to me. There's this chain somewhere? Are there links? Is it like a bracelet? What's going on? And then there was even more confusing. They would have this staircase drawn in the textbook, and that would show the electrons bouncing down the staircase. And that made no sense to me. I was like, what is going on here? Okay, so the electron transport chain, you know what it is? It's a series of proteins embedded in this membrane right here. That's all it is. This membrane here, this inner membrane of the mitochondria, is just like any other membrane. It has a phospholipid bilayer. But embedded in that bilayer are some very special proteins that are able to transport electrons down themselves, across, right? So let's say this is an electron transport chain here, and if I were to enlarge that, and I will show you an animation of it somewhere else because it's hard to draw this, but there are electron transport proteins that are like all in a row. There's like 4 or 5 of them. They're all through this membrane. There are hundreds of electron transport chains, or a consecutive group of special membrane proteins embedded all through this membrane. That's what the chain is. It's a chain of embedded transport proteins in that inner mitochondrial membrane between the matrix and the intermembrane space. They're all through there. Those little blue dots that I'm drawing are the electron transport chain. So let's just leave it blue, all right? So that thing there, that thing there is an electron transport chain. Electron transport chain. Let me do it. Electron transport chain. Okay, I'll draw it then. There. And all it is a series of embedded proteins that catch electrons and push them along the way. So here's what's going on. The electrons are in the NADHs and FADHs, right? And they're going to carry those electrons over to the first protein in that chain. Those electrons are going to flow down the chain this way. They're going to go, they're going to go reduce those proteins and then get oxidized, and then reduce one and then get oxidized, and flow down and lose their energy as they go from protein to protein in the chain. So what? Okay, so what happens is, as a result of that, these special proteins, these electron transport proteins, they can do two things at the same time. They're very good multitaskers. They are able to have electrons flow one way while they push hydrogen ions another way. So there are also always hydrogen ions floating around, and I'm going to draw them as little plus signs, okay? Little plus signs. Those are hydrogen ions. And basically all a hydrogen ion is a proton. So as electrons go down this transport chain, you are pumping hydrogen ions into the intermembrane space. I don't know if you remember the cell transport unit we did. Remember when we talked diffusion, right, and gradients? There is a gradient being created here. Literally a proton gradient, because the more electrons are harvested from the glucose and are given to the electron transport chains, the more hydrogen ions, and really these are hydrogen ions, but I'm just using shorthand and calling them little plus signs, okay, get built up over here. And that is creating a proton gradient. A proton gradient, meaning that there is more protons or hydrogen ions over here in the intermembrane space than there are in the matrix. Nature doesn't like that. Nature doesn't like to have something more concentrated in one area than it is in the other. It's always going to try to allow things to go down their gradient. This is like active transport, kind of. It's using the electron's energy to pump those protons into the space. It's kind of like pulling back on a spring. So now the final end of the story is that along with these blue electron transport proteins in this membrane, there is also, I'm going to make it purple, there is also this really, this very powerful, I guess you could call it a synthase protein. I'm making them purple. These little purple things that I'm drawing in here are the only way, they're actually a transport protein, it's the only way that the protons can get back through, back across the membranes. Remember, protons can't handle going through phospholipid bilayers with that hydrophobic sandwich of tails in there, right? So it turns out that those purple things are called ATP synthases. And what does an ATP synthase do? It synthesizes ATP, hence the story's end, right? Isn't this exciting? Okay, so we've got the protons that now, because they've built up over here, they've built up over here in a gradient, and their only way back through is through the ATP synthases, so back they go, right? back they go. And it turns out, I'm going to have to make this larger, but it turns out that here is an ADP molecule hanging out right close to that ATP synthase, and there's always inorganic phosphate hanging out as well. And the ATP synthase, every time hydrogen goes through it, it makes ATP. And I can show, the animation is a lot clearer than this, but that's the basic gist of it. Look at how many hydrogens are over here. And every time they flow through ATP synthases, which are also all over the place, you make ATP. That's why you can make so much more ATP using the electron flow and the electron transport chain than you can with anaerobic respiration, where all you've got are little tiny enzyme reactions that are putting phosphates onto ADPs. This is called oxidative phosphorylation, meaning that you're using the oxidation of glucose, ripping electrons off glucose, which is oxidation, and you're using the power of those electrons to stick a phosphate onto ADP and make lots of ATP. Okay, where the oxygen comes into all this, my picture is getting a little bit chaotic, but the oxygen is coming into all this. It'll be a lot easier on the next video when I show you the video, but the oxygen comes in here. I'll make it brown. So oxygen gas, remember, is small and nonpolar, so it can diffuse right through and go into wherever it needs to go. And when the electrons reach this point right here, when the electron reaches that point, it's got to go somewhere. If it stays there and oxygen doesn't come and pick it up, right? If oxygen doesn't come in and pick up that electron and become water, then that electron kind of gets stuck. And then all the electrons behind it also get stuck. And that turns off the electron transport chain. If you have no oxygen to come in and be the final electron acceptor of all those electron transport chains, it brings all of the pumping of hydrogens to a halt. And if you can't pump hydrogens, the hydrogens can't move through the synthases, and you can't make that big ATP yield. Okay, that was a lot. That was a mouthful. But that's basically it. That is aerobic respiration. That's what's happening in 1 billion mitochondria in all of your cells, particularly in your muscles that have a lot of them, and in your brain and nerve cells that have a lot of these. This is what's happening. That's how you're making most of the ATP, is by these electron carriers, NADH and FADH, coming over here, right? And remember, these guys can come over here too, and so can these. These are going to come in and help make ATP as well. And as long as you have oxygen to come in and keep it all going, you can make a lot of ATP. Okay. Before anything else, I'm going to share an animation with you that explains in particular this area here, which is kind of chaotic. And the animation that I'll show you is much more clear, okay? Let me show you that. And you also have this same animation. You could go see it anytime. Okay. So this is the same McGraw-Hill animations, and we already saw glycolysis. We're going to go straight for the electron transport chain. Come on. Or not. What's going on? It's dead. Of course it is. Okay. Oh, wait. Okay, my computer has just frozen. Completely frozen. I can't even go back to you guys. That's interesting. Okay, I'm going to have to leave and come back, guys, because everything has just completely... Are you guys there? Are you guys there? Yes, I can hear you. Okay, coming in. My screen is frozen. I can't move. I'm going to have to log out and log back in. Just hang in there. Hang in there. I don't know what's going on. Okay, are you there? Sorry about that. No, I think the setting turned off. Oh, good. Thank goodness. All right. Okay, I'm sorry about that. It just completely froze up. All right. Okay, I'm going to try to show that animation again because I think it would be worth you seeing, but I'm gonna have to, oh, geez. Let's see if it'll do this without shutting down again. I think BBCU is freaking today. It's doing all kinds of really strange things. Okay. Go into classroom and go from there. All right, this is your classroom. Right. Okay, so if you go all the way down to the bottom of this list, it says useful YouTubes. So if you're looking for something to help you study, here they are. All Mr. W's songs and things are there. Here's the McGraw-Hill here animation. Let's see if it'll work. Allow. Yes, allow. Okay. Let's see if we can get it to. There we go. Okay. All right. So this, I think, is the right one. Let me just make sure that. Yeah, this is a better one. All right. This is the mitochondria, and I'm just going to let this play. Oh, wait. Can you see it? You can see it, can't you? Yes, you could see it.
Oxidized during glycolysis and the Krebs cycle. The coenzymes are reduced to NADH plus H+, FADH form. In mitochondria, oxidized from NADH plus H+ to electron carrier proteins, and the proteins are transferred across the membrane.
Okay. It's hard to hear. Yeah, I can redo that. Yeah, I think I can get it. I think I can get it better now. Hold on. Share audio this time. Yeah. Okay, here we go. There. All right, so it's going to, let me just go backwards for a minute without the sound. All right, so there's the mitochondria. When I said electron transport chain, look at the small area that they're showing here. Here's the matrix. Here's the intermembrane space. There's the membrane that is between those two areas. And that's where those proteins are embedded. So if you go a little closer, there they are. This is the electron transport chain. Here, these blobs are proteins that are sitting in that membrane inside the mitochondria. And they are accepting electrons here and here. And the electrons, if you look, I'm going to let it play in a minute, they're flowing this way, right? The electrons flow this way. So the NADH brings its electron, releases it to this little pink thing, and then the electrons flow this way. While the electrons are flowing this way, these guys are multitaskers. They're pushing hydrogen ions from here to here. So hydrogen ions are going this way, right? And in the intermembrane space, you're getting this buildup of hydrogen ions. And then this purple thing is the ATP synthase, which is their only way back in. Okay, I'm going to turn off my sound so you can hear it better.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H plus and FADH2. In the mitochondria, the electrons from NADH plus H plus are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
Okay, so that is a little bit clearer image of what's going on with the electron transport chain. That's a lot of information, but that's pretty much it. That is what you need to know in terms of understanding how the energy from glucose is really being harvested, and then eventually how it gets to make all that ATP in the presence of oxygen, in the presence of a mitochondria. Is that clear to y'all, or do you think you could get it if I record this and post it for you? You're going to be able to do it? You're going to be able to finish it? Yes? Okay. So I'm giving you the rest of class to work on that, on finishing that animation that you started last time, the one that's kind of like the Wiley tutorial. So that's a really important must do. So you should have enough now to finish parts 3 and 4, which are about the Krebs cycle and electron transport chain. I also have the mastery quizzes are up on Albert. That's the last thing that you need to do, and it's really good preparation for the test on Thursday. The test on Thursday, let me just turn this off